I just want to say it's a great pleasure to be here. My name is Vicki Grassian. I'm from the Department of Chemistry and Biochemistry at the University of California, San Diego. It was nice to hear about some of my colleagues from the past, Leslie Orgel, Stanley Miller, who all worked on some of the great chemistry early on, and that you're talking about at the symposium. So today I want to talk about some of the work that we're involved in having to do with the surface and multiphase chemistry / photochemistry of pyruvic acid and pyruvate. And we haven't heard too much of these molecules, and I was trying to stare at Nick's really small molecules. I don't think he had it. He had, I think it was a glyoxylic acid and not pyruvic acid. So let me just introduce pyruvic acid. It's the simplest alpha keto acid involved in several biochemical pathways. That's not going to be my focus today. Some of you might be studying these different pathways, but it's not my focus. What I want to talk about today is just some different aspects of pyruvic acid. First of all, you may not know it's also an important atmospheric alpha keto acid. It's formed in the atmosphere via oxidation of isoprene, so a volatile biogenic compound from trees. The other thing you might not know is that it is one of the dominant gas phase carboxylic acids detected in occupied classrooms. What I mean by that is we are emitters of pyruvic acid into the gas phase. Another thing I just want to point out, because we'll be talking about the photochemistry, is that there's really interesting, very different photochemical pathways for gas phase versus aqueous phase pyruvic acid. When we think about the photochemistry, I'm mainly talking about the chemistry that overlaps with the solar spectrum, so anything with wavelengths greater than 300 nanometers and above. And this, in the aqueous phase, occurs at around 325 nanometers and is due to the N-pi to pi star transition localized on the C double bond O, the carbonyl group. So on the right-hand side of this slide, I show you the lowest energy conformers of gas phase pyruvic acid. One is called the trans-cis conformer, the TC conformer. And in this particular case, what you can see is that you have a hydrogen bonding interaction of the OH group from the carboxylic acid group pointing toward the ketone. So this is a 3-carbon system. So you have the methyl group, the carbonyl, and then the carboxylic acid. In the case of the TT isomer, or the conformer, excuse me, we see that the OH group is pointed away. So these are fairly close in energy, about 2 kcals per mole difference, but we do have these 2 conformers. So the question that we were asking, we had a few of them, but what about what happens when pyruvic acid is absorbed on a surface or in the presence of salts at high ionic strength in solution, representative of marine environments, and you'll see in a little bit why we're interested in marine environments. So really what I want to do is talk about surface and multiphase reactions of pyruvic acid. We heard a lot of very interesting surface chemistry just a few minutes ago. In fact, fascinating surface chemistry in my opinion, where we have these solids, sometimes these dissolve, sometimes they don't, but reactions are occurring often at the surface of these solids. I'm going to be talking about oxide surfaces. We heard a little bit about oxide surfaces from Tom just a moment ago. But, you know, as he just said, you know, what are the reactive surface sites? What's the speciation of different things that are on the surface? Very interesting, you know, surfaces can really provide high concentration environments, especially when you get to high coverages. And there's also the catalytic and photocatalytic aspects of surfaces that are so interesting, and we just saw a moment ago form such important compounds related to prebiotic chemistry. I'm also going to talk about some multiphase reactions. And, you know, these definitions of surface or heterogeneous and multiphase chemistry, I took from Ravi Shankar's paper that came out quite a few years ago now. And so what we're talking about is reactions that occur in the aqueous phase. Sometimes we think about aqueous aerosols. I'll be basically showing you some bulk aqueous phase, but what's interesting about aqueous aerosols, these are micron-sized droplets You have reaction, you have diffusion. There's been a lot of talk lately about reaction kinetics being enhanced many orders of magnitude in the aerosol, the aqueous microdroplet, due to fast diffusion and surface effects. Graham Cooks just recently wrote a really nice annual reviews of physical chemistry on this topic. There's interest in what's the speciation at the air-water interface versus the bulk. Again, you can have very high ionic strength environments in these aerosols. There might be some alignment effects. People have talked about chemical gradients as well. I'll just touch on a few of these today. So I'm going to talk about three things here in my talk. I'm going to start off with just discussing pyruvic acid adsorption and photochemistry on hydroxylated SiO2 surfaces. So we're going to be introducing pyruvic acid from the gas phase. We only have to worry about this species, although we saw we have different conformers of that species. Then I'm going to be talking about pyruvic acid adsorption, its chemistry, its photochemistry on other oxide surfaces, I would say more reactive oxide surfaces, such as aluminum oxide and titanium dioxide. And then I'm going to talk about the role of what I call environmental factors in the aqueous phase, marine relevant multiphase chemistry of pyruvic acid. And what I show you here is that things get pretty interesting pretty quickly when you go into the aqueous phase, because we have to worry about four forms of pyruvic acid. First of all, the protonated and deprotonated forms, but you also get the enol form as shown here. And what's interesting is that the enol form is a dark form, if you will, and I'll talk more about that later in this talk. Okay, so we have several overarching questions. How does adsorption of this very interesting, important molecule impact its structure, its speciation, its chemistry, its photochemistry, even just the protonation state when you have something adsorbed is interesting. What about these different conformers? Do we have one over the other? And is this adsorption reversible or irreversible at 298 Kelvin? So one of the differences in the talk I'll be giving today, we're not heating up to higher temperatures as we saw in Tom's talk to initiate chemistry. And we're using in situ vibrational spectroscopy as a probe. So you're going to see a lot of spectra. So here, just looking at the gas phase pyruvic acid, the spectroscopy and structure. I've already discussed with you, there's two conformers. Here we see the gas phase spectrum. In the gas phase spectrum, we can see in the OH stretching region evidence for these two different forms, the TC and the TT form. What I want to tell you is all these calculations that I'll be talking about today were done by my collaborators, Marie-Pierre Guéchou, Marie-Laure Zulpisi, and also Dominika Lesnicki, who was a postdoc of Maria Lore. So I just wanted to tell you they did the calculations. So we see that these exist in the gas phase. Now let's adsorb this molecule on a surface. And so what I'm doing here is just showing you the infrared spectrum, in situ infrared spectrum, as we change the gas phase pressure of pyruvic acid and its interaction with hydroxylated SiO2. So on the left, we see the whole spectrum over a wide range from about 1,200 or so, 1,300 wave numbers to 4,000 wave numbers. A couple of interesting points. One is that you start to see hydrogen bonding with SiOH groups. That's the 3744 wave number peak that goes away. So these are different spectrum. And then you see this peak at about 3,400 wave numbers due to the hydrogen bonding. So from isolated SiOH groups to hydrogen bonded. And then you also see bands in the spectrum due to pyruvic acid. We can see some large changes. If I go back to the gas phase spectrum, here the most intense peak is at 1805 wave numbers, but we see that we don't even have anything at 1800 wave numbers, and that band has actually shifted quite a bit towards 1776 wave numbers. The other thing I want to note is that when we evacuate this gas phase, we have that dotted line, and really pretty much everything desorbs from the surface, and so we have this reversible molecular adsorption. So what about the structures on the surface? So again, our collaborators helped us out with this, doing some MD simulations. And so again, on the left, what we're showing is the gas phase structures. And then on the right, we start to see that we do have structures that are associated with the TC and the TT formation, but they bond differently. So the TC conformer binds through the C double bond O of the carboxylic acid group, and then we have another hydrogen bond between the OH group from C double bond O H with the surface. And so you can, hopefully you can see some of these hydrogen bonding interactions. And so this is the surface. We simplified it just to look at the different interactions with these hydroxyl groups and the pyruvic acid. Here we have another structure where we see C double bond O interacting with the surface SiOH groups, and then we see the carboxylic acid group interacting with the oxygen of the SiOH. One other interesting thing that we saw was that the dimer was pretty stable on the surface, and we have evidence of that also in the infrared spectrum when we start to deconvolute that very intense C double bond O stretch in the infrared spectrum. So we see we have the dimer. So, you know, that's what's interesting about these surfaces. They can orient these reactants, if you will. Here we have this sort of double hydrogen bonded interaction with the carboxylic acid groups. And what we were able to do was see that that dimer increased with surface coverage. And here's some of the binding energy calculations that we were able to obtain from the calculations. One of the questions we asked was what happens in the presence of water? We've talked a lot about hydration, dehydration, rehydration today, and so we're always interested in how water impacts surface chemistry. This is showing a vibrational spectrum as a function of increasing relative humidity. And the thing that what you want to see is as we increase relative humidity from dry, so less than 1% RH up to 81% relative humidity, we have an increase in this band at 1627 wave numbers due to the bending mode of water absorbed onto silica, and at the same time we see a decrease in the 1737 wave number region where we have that associated with the pyruvic acid, as well as decreases in other spectral regions as well down here at 1355, 1389, 1428 wave numbers. So we see that water competes for some surface absorption sites and actually decreases the amount of absorbed pyruvic acid. But we don't see any pyruvate forming. We're not initiating a proton transfer, if you will, reaction on this surface. We had our collaborators also look at this, and what I want to sort of point out is that what we were able to determine through the calculations was, now we're looking down at the surface, and the pyruvic acid is really above everything else, and there's a water between the pyruvic acid and the silica, and it's that hydrogen bonding between the water, the silica, and the water and the pyruvic acid that is what's forming when we have a lot of water on the surface. So we sort of displace the pyruvic acid up, and we see that water is interacting as that bridge between the silica surface and the pyruvic acid. So some interesting observations and what we can learn from these MD simulations. We also looked at the photochemistry in the presence of the silica surface. Here's the surface difference spectra we see as we irradiate with a solar simulator, a decrease in the amount of pyruvic acid absorbed on the surface. We can then also do look at the rate of pyruvic acid loss in the gas phase. We see some differences in the presence of silica. We actually see it slow down a bit, but we really didn't see anything that was unique in terms of products. We did see a little bit of gas phase acetic acid, which had been seen before. So now let's compare that to another surface. The other surface that I want to compare it to is that of what I would say is a more reactive surface, namely hydroxylated Al2O3 aluminum oxide and titanium dioxide. So let's look at that. So here's some very similar experiment, pyruvic acid absorption from the gas phase onto a hydroxylated aluminum oxide. I call it a more reactive surface. And there's a few things that I should point out here. Again, we have some reaction occurring or interaction occurring with the hydroxyl groups on the surface. But what was really interesting is that we start to see absorption bands 1680, 1655, 1607, even 1470, all in this region that we saw nothing before when we looked at absorption on silica. And what we see is that we are starting to form pyruvate as one of the dominant species in the spectrum that we're observing right here. And what we're getting is reaction of pyruvic acid. And this happens with a lot of carboxylic acids. And I think you even showed some of this, Tom, in your talk, where you can react on the surface. We can react with hydroxyl groups, and we can actually form and absorb pyruvate. What is interesting about this is that if you look at the black dotted line in these spectra, and sorry if it's hard to see or not, but really we maintain a lot of the surface absorption, and we see that there's a great deal of irreversible absorption. And in a moment, I'm going to show you that we even make some higher molar mass species that we've been able to detect with mass spectrometry. But before I showed you that I just want to show you that TiO2 is similarly reactive. And here we're just looking at, we had set the gas phase pressure. We look at how the bands grow in and as a function of time. And we see something very similar when we pump out the gas phase. We get the dashed spectrum shown there, showing that this is staying a lot strongly bound to the surface. We also propose that pyruvate is the main form, but again, we form these other products as well. And this paper was published in the Journal of Physical Chemistry. I guess it was in 2019, the good old days when we used to see each other. And some of my students, Mike Alves and Yuan Feng, was involved in this work, as well as Veronica Vita, who's motivated a lot of our work on pyruvic acid. And she's done some great things with that. What we did then was we looked at, can we get even more chemistry going? Here we look at the photochemistry of pyruvic acid on aluminum oxide and titanium dioxide. Again, we're looking at different spectra, but now instead we start to see even more products forming. Those are the positive bands. I won't go through the detailed analysis of these. It's actually complicated. We see that we get chemistry going on. We're looking at the first order decay of the pyruvic acid, or that 1736 wave number peak, 1741 in TiO2 of pyruvate as a function of time. Again, we see these other bands growing in, so we have these new products. And it was just going to be too hard to say anything more with IR spectroscopy. So what Mike Alves did when he joined the group was extracted these products and did some high resolution mass spec analysis. And so what we see is that we get these higher order, these oligomers, these larger molar mass species, if you will, just by simple absorption on the surface under very mild conditions of room temperature, 298 Kelvin. And then if we have solar radiation, we even get more products. So we formed zymonic acid in the dark or in under light conditions, but we formed two products, parapyruvic acid and 2,4-dihydroxy 2-methyl-5-oxohexanoic acid, MOA, both when we have the presence of light. So really starting to get some chemistry going. I know we didn't talk about this too much yet. How these the solar energy and other forms, gamma rays, other high energy particles are out there that can also, these photons, these particles can also be initiating a lot of chemistry. So let me just conclude from this parts 1 and 2, if you will. I think we've learned a lot of interesting things about a relatively simple 3-carbon system, pyruvic acid, and how we can be able to look at how these conformers are stable on the silica surfaces, but we also get the dimer. In the case of some more reactive surfaces where we have more basic hydroxyl groups, we get pyruvate forming, these other that can go on to form other products as well. Water plays a role. We didn't do any of this rehydration, dehydration experiments, but I do find those fascinating. But we see that as a bridge. And then again, we form these other compounds. So that's just some of the interesting experiments that we were able to do for pyruvic acid on oxide surfaces. The next thing I want to look at is, just trying to see where I am. Okay, I'm doing great. Okay, I'm doing better than Tom in timing. Only kidding. Only kidding, Tom. Okay, so the next thing I want to talk about is the role of environmental factors, particularly salts and pH in marine relevant multiphase photochemistry. And I sort of told you already, we kind of start off now with like 4 different forms, and we'll go through some of that. So I just want to step back and say the work that we do on marine systems is part of another CCI, the Center for Aerosol Impacts on Chemistry of the Environment. We call it CASE. It's a center that's been around for quite a while. I mean, phase 1 was started over 10 years ago, and those who've written these proposals know what I mean by phase 1, phase 2A, phase 2B. We're in phase 2B right now. We have a few more years. This is a giant picture of the group. We used to meet in San Diego all the time, so this was when we could all get together. It's really hard to see, but in the middle, I think is in the white, is Kim Prather. She's the director of the center. She's done a great job. I'm next to her, I believe, and I'm co-director of the center. And I know that your center has played a big role in mentoring our center, and we really appreciate that from so many different perspectives, because we started after you. So thank you for all you've done and all your encouragement and your insights into how to run one of these large centers to get the most out of the opportunity that we all have through that CCI program. So I'll be talking about some of the photochemistry aspects of what we're interested in. And it's not just the aerosols. We're actually interested in the ocean, atmosphere, system, from the seawater to the sea surface microlayer, that top layer, to sea spray aerosol that gets out into the air. Okay, so we want to discern the role of these environmental factors. And there's so many interesting questions one can ask, you know, the role of solvation, salts, ionic strength, monovalent versus divalent cations, solution pH. So I list marine relevant solution pH from 2 to 8. People are going to say, well, what is she talking about? She's crazy, pH 2. But I have something I want to show you today to say why we can, we think that's a relevant, marine relevant pH. And, you know, I'm not going to talk too much about this today, but the role of interfaces are so interesting, that air-water interface. I hadn't worked on the air-water interface before in my career, but we've done some things now. And there's really the propensity for the protonated form at the surface. And this chemistry is really driven by the surface activity of the protonated form relative to the deprotonated form. And so we've studied this in some detail. And this is for non-noic acid, and I just reference our ChemSci article. Sorry, I have some dogs with me, and they were just sleeping a moment ago. I guess I got them excited about something when I said interfaces. Okay, so marine relevant pHs, I say, go from 2 to 8. Well, how can she say that, one would ask? And so here I'm just showing you some of our newest results from a study we did in summer of 2019, where we used Scripps Institution of Oceanography, the hydraulics lab, where we have an ocean atmosphere system where we can actually break waves, and that's what was shown here on the bottom. We have this beautiful picture from Nigella Hilkarth showing a breaking wave, and then we were capturing the sea spray aerosol. And this time when we did this kind of experiment, because we've done these mesocosm experiments before, we decided to try to capture and measure the pH. So here we have the bulk pH of seawater at about 8. We have the sea surface microlayer, which is a little bit lower at 7.8. And then we have the pH of the aerosol. So how did we measure the pH of the aerosol? We measured the pH of the aerosol by simply depositing this onto pH paper. Okay, we did the simplest experiments in the world, and we got a PNAS paper. Okay, so that's kind of cool. But what we saw was that really within minutes, we were seeing acidification of the aerosol. And even though I have this as pH of size of 1 to 2 microns, this is pretty much the supermicron, the largest size particles. We see that at about pH 4, and these smaller particles are closer to pH 2. And we talk about this in great detail in this paper that just came out a few weeks ago this year. Okay, so I'm just giving you some reasons why we could study those pHs. All right, so no, okay, stop. Okay, so here we're just looking at some of the optical properties, namely just the UV-vis absorption spectrum as a function of pH and in the presence of salts. Again, we're looking at the 4 forms on the left-hand side, and I want to remind you that the diol form is dark, okay? More diol, less chromophores, if you will, absorbing wavelengths that we're interested in, which is above 300 nanometers. So we do this experiment under conditions where we mostly have pyruvic acid, the protonated form of pyruvic acid, as well as the diol. And then we do these experiments under conditions where we have the deprotonated form. And so what I want to show you here, in the presence of salts or just water, we have very little changes in the absorption maximum. But in the case of pyruvate, when we have calcium present, we have a shift in the wavelength shown there. So we talked to our collaborators. On this time, our collaborators are Benny Gerber and Dorit Shemesh, who were calculating the excitation of pyruvate in the presence of water, in the presence of calcium. And you see that green is the calcium, which binds to the carboxylates. And what they were able to do was show in their calculations that you get the shift that we observe. If you look at the blue versus the red, what we observe in terms of the effect of that binding, that divalent cation binding. And so we were able to see that shift, and we were able to see some in both the experiment and in theory. Again, if you look at the system, we start to probe all the species that are formed in water, or when you have the presence of this divalent salt, and we increase, we show this here. And if you look at things in a very subtle way, you can start to see changes in the relative amounts of enol, the diol, I should say, versus the ketone. The neutral form is the protonated form, or the pyruvate. And so all those are showing changes in the presence of calcium. The other thing we showed on the bottom part is the fact that we could map out the UV-vis spectrum just based on how much of that ketone versus the diol form that we have present. So we really show that it's the light absorbing component that's interesting. We then, looked at some photochemistry. Again, here's the shifting that we see in the overlap. We're looking at the spectral irradiance on the left. When we compare the concentration of the pyruvic acid or the pyruvate as a function of time, we see that the calcium binding to the pyruvate, in fact, has an influence on the the rate of the reaction. So we see a little bit of the photodecay being suppressed in the presence of calcium, but we don't really see differences in the products that form. And the aqueous phase chemistry has been studied quite a bit by Veronica Vita. But we wanted to look specifically at the effect of calcium binding. We see some differences in the photodecay, but not much difference in the products that form. So, we really wanted to start to look at, and we're doing a lot more than this, that of course I don't have time to tell you today, but really trying to understand the roles of things like salts and divalent calcium binding. We're really interested in looking at the impact of pH on some of these reactions, on these chromophores and how that might change their absorption spectrum. And there's a lot more that we're doing as part of that center, but this is just some of the things that I wanted to show you today at this symposium. So I hope that I've talked and showed you some interesting chemistry having to do with pyruvic acid in surface reactions and in aqueous phase. And I just want to end by thanking my the wonderful people that I work with, both collaborators as well as graduate students that were my group, like Yuan Feng, Mike Alves, Kristen Wall was a postdoc, and then we've been working on this aqueous phase chemistry with graduate student Man Liu and others in my group, as well as collaborator such as Dorit Shemesh and Benny Gerber. So with that, I'll end my talk, and I'll stop sharing. Thank you.